Dear students, welcome to my class today. We will study what is Genomic DNA library, how it is prepared, and what are its uses. DNA is hereditary material. It contains genes which are transmitted from one generation to another generation. So, to extract the DNA and prepare this genomic library is useful for researchers and for genetic engineering program. What is genomic library preparation? Entire DNA of an organism is called genome. Now that DNA is broken up into pieces, and those pieces, they are multiplied, then they are stored in the form of a library from where researchers or genetic engineer can pick up the gene they want to study, and this is also used for sequencing the DNA. So what is genomic DNA library? Genomic library is a collection of recombinant DNA within transformed cells in which gene is present in a single copy. Now, what are transformed cells? Supposing we have inserted that fragment of DNA into a vector, plasmid or phage, then this is called as transformed cell. What are the uses of genomic library? Entire DNA of an organism can be sequenced. I mean, we will come to know A gene comes after B, B gene comes after C. Whole sequence will be known. And by knowing the sequence, we can know the position of the genes. We can also know various functions of the genes. Then those genes, where they're required, like in gene therapy or in genetic engineering or in production of genetically modified crops, it can be used. And very important aspect of this is Human Genome Project. What are the steps for the preparation of genomic DNA library? First, we have to isolate the genome, or we can say entire DNA of an organism. For that, we have to treat that with the alkali. So this is called as alkaline lysis. The alkaline lysis is used for extraction of DNA. This is a very fast, reliable, and clean method to get DNA. Then, after getting DNA, now that is treated with the phenols. Now, phenols are used for removing the proteins which are associated with the DNA. We know that when we study the structure of DNA, DNA is associated with acidic and basic proteins, histones. Then those proteins will be extracted with the help of phenols. After extracting the DNA, removing the proteins, then we can break that DNA into pieces. Now, those pieces can be done either by sonication or by restriction enzymes. Restriction enzymes are basically highly specific enzymes which can break the DNA at specific places. So, fragments of DNA will be generated. Another method is sonication. The sonication is, you can say, sound energy is used to agitate particle or sample. When DNA fragments can be agitated by sonication. Then another technique is by pipetting or mixing. So after getting DNA fragments, we can multiply those DNA fragments with the help of vectors. For multiplying DNA fragments, we will use the vectors which can accommodate big pieces of DNA. So we'll use large capacity vectors for eukaryotic genome. Now, for cloning, what we need to have vectors like lambda phage vectors or cosmids. So what we will do, we will cut the DNA of lambda phage vectors or cosmids, and after cutting that DNA with the help of restriction enzyme, we will add that DNA which we have fragmented by isolation of whole DNA, then pieces of DNA have been created. They will be recombined with the viral vector. Now, the viral vector DNA or cosmid vector DNA, then they can be packed inside the phage or they can be packed inside the plasmid. Then multiplication will take place. Many copies of that DNA will be created. Those copies will be similarly with the DNA which we have generated by fragment. So multiple identical copies are called as clone. This is called as cloning of genomic DNA. Various enzymes are used in the whole process, which play an important role in the preparation of genomic DNA library. One are restriction enzymes. Restriction enzymes are very specific enzyme. They will only cut at specific places on the DNA. Then another kind of enzyme is alkaline phosphatase. Like we are using certain vectors, we are using plasmids. When we cut the DNA, that has a tendency to re-anneal itself. Actually, when we study the structure of DNA, there is 5' end of the DNA and then 3' end of the DNA. 5' end has phosphate, 3' end has OH. Now, if phosphate is not removed from the 5' end, again DNA can re-anneal itself. So to prevent re-annealing of the DNA, because we want to insert our DNA fragment into the vector, so we will treat the 5' end with alkaline phosphatase so that from the 5' end phosphorus is removed. Now when we have done this process, then we will insert our DNA fragment into the vector. The vector DNA and DNA fragment will combine with the help of enzyme DNA ligase. The function of DNA ligase is gluing of the DNA fragments. Now, when vector has taken the DNA fragment from the outside, this is also called as recombined DNA. So, we'll take circular plasmid. Circular plasmid is extracircular DNA present in the bacteria. Now, that can replicate independently, can form copies. Now, that DNA is digested with the help of restriction enzyme. After restriction digestion has been done in treatment of the plasmid DNA with the restriction enzyme is called as restriction digestion. So we will remove its 5' and phosphorus with the help of alkaline phosphatase. This is called as alkaline phosphatase digestion. After taking this alkaline phosphatase digestion, linear plasmid is created. Now linear plasmid DNA now can be combined with fragments of DNA which has been obtained from the genome of a particular organism. Whole genome of a particular organism has been broken up into pieces. Now that will be recombined with this vector or plasmid. Now recombined DNA along with the plasmid can be stored. Now whole set of the genome has been broken up, supposing into 10 pieces, then 10 pieces will be stored in different vectors. One vector will have one kind of DNA. Now, if we say entire genome has been broken up into pieces, like if we talk about Escherichia coli, Escherichia coli is a bacteria. Whole DNA of Escherichia coli will need 1500 cloned fragments to accommodate the genome of Escherichia coli. Similarly, if we talk about yeast, yeast is a unicellular fungus where 4600 clone fragments are required to accommodate the genome of yeast. And then human beings, 8 lakh cloned fragments are required to accommodate the genome of the human beings. So in this way, we have prepared a library from where we can pick our gene of interest. Now that gene of interest can be used for various purposes. We want to know the sequence of the DNA, or we want to use them for the genetic engineering experiments. We want to use those genes in the research process. We want to those fragments of DNA in gene therapy. Wherever we want, we will pick up those fragments of DNA and we will use in our various interests. Thank you so much. I hope you enjoyed my class. If you like my class, then please subscribe my channel. You will get more information on this channel. Thank you.